In this video example, we discuss a GNPTAB frameshift variant for eligibility towards ASOs. First, we begin with a variant check, where we assess if the variant is correct. More detail on how to check if the variant is correct can be seen in the ABCA4 video example. In this case, the variant description is correct. At this step, we could also check if an ASO exists. On PubMed, I search up the gene name, variant, as well as different terms for ASOs. In this case, I use ASO or antisense. More search terms can be found in our guidelines. Once we click enter, we are directed to this article by Matos et al., titled Development of an Antisense Oligonucleotide-Mediated Exon-Skipping Therapeutic Strategy for Mucolipidosis 2, Validation at the RNA Level. As suggested by the title, the validation is only at the RNA level. Therefore, validation on the protein level is still required, so we cannot classify this variant as eligible according to our guidelines. Therefore, we continue to proceed with analysis as usual. Scrolling down, this article provides us with useful information. We see that this variant is loss of function and that the variant is found in exon 19. This article provides us with the information for step 2. The mechanism is loss of function. We still need to check the inheritance pattern. As suggested by our guidelines, we can use OMIM for this. The inheritance pattern is autosomal recessive. Given that this variant is a frameshift variant and no splicing data is available, we can skip over step 3 and instead jump to step 4 as suggested by our guidelines. Since our variant is loss of function and autosomal recessive, we can assess this variant for exon skipping. Let's begin by assessing whether the variant is in the first or last coding exon, if the exon is out of frame, or if a stop codon is generated if skipped, as well as checking the size of the exon. Note that we have another exon skipping example, where we assess the eligibility of an exonic single nucleotide deletion in the CTSF gene for eligibility towards exon skipping. The CTSF video provides a few other strategies on how to go about assessing whether we can use exon skipping ASOs. In this video, most of the information can be obtained from the paper by Matos and team. In Figure 1 from the Matos paper, we see that this variant is in exon 19, and it is not in the first or last coding exon. We can also see that this deletion results in an out-of-frame transcript, and that skipping exon 19 brings the transcript back into frame, since exon 19 is in frame. In panel B, we see the amino acid sequence of the transcript without exon 19 in the second row. A stop codon is not formed. Lastly, in the figure description, we see that the fragment coded for by exon 19 is 56 amino acids. Using UniPro, we can see that the total length of the protein is 1,256 amino acids. Therefore, we are only losing about 4.5% of the total protein length, less than the 10% threshold mentioned in our guidelines. Lastly, we want to assess whether this exon codes for an important domain. Again, other strategies are discussed in the CTSF example. For this video, we will be using the Matos paper. We can see here in the discussion that exon 19 codes for the stealth domain, and that only 3 missense mutations are found in the human gene mutation database for this domain, of which only 1 is associated with a mild mucolipidosis phenotype. If we scroll down, we also see that in silico and in vitro analyses of missense variants did not find any important amino acid residues in this domain essential for phosphotransferase function. Therefore, the authors conclude that this domain is less important for phosphotransferase activity. We know that our variant is in an in-frame exon, is not in the first or last coding exon, and does not create a stop codon when skipped. We also know that the exon codes for about 4.5% of the coding transcript. However, the exon does code for a functional domain. We see here that exons that code for a functional domain are to be classified as unlikely eligible. Therefore, we classify this variant as unlikely eligible for exon skipping ASOs.